Hello, this is chapter 27 on prokaryotes. Biologists currently divide all organisms into three groups called domains. Domain bacteria, domain archaea, domain eukarya. The organisms making up two of the three domains, bacteria and archaea, are called prokaryotes. The domain eukarya include four subgroups: protists, plantia, fungi, and animalia. This chapter will examine first the structural and functional adaptations that contribute to prokaryotic success, genetic diversity in prokaryotes, diverse nutritional and metabolic adaptations, crucial roles of prokaryotes in biosphere, and beneficial and harmful impacts of prokaryotes on humans. Due to their ability to adapt to diverse habitats, prokaryotes are the most abundant organisms on Earth. Some prokaryotes thrive almost everywhere, including highly saline environments, like Dead Sea in Jordan, Spanish Lake, shown in the figure below. The color comes from trillions of living prokaryotes. Those organisms are called halophiles. Some thrive in very hot environments. We refer to them as thermophiles. For example, some archaea live in sulfur-rich volcanic springs as hot as 90 degrees centigrade. One extreme thermophile that lives near the deep sea hot springs, informally known as quote-unquote 121, since it can reproduce even at 120 degrees centigrade. The color you see here comes from trillions of living prokaryotes. The pigments are found in the cell wall of these organisms. Other prokaryotes can survive 3 million rads of radiation, 3,000 times the dose fatal to humans. Some can grow at pH of 0.03, very acidic, enough to dissolve metal. Some found living in rocks more than 3 kilometers below Earth's surface. Let's start with the first concept. Structural and functional adaptations contribute to prokaryotic success. Prokaryotes were the first organisms to inhabit the Earth. Most are unicellular, although some species form colonies. Most prokaryotic cells are 0.5 to 5 micrometer, much smaller than the 10 to 100 micrometer of many eukaryotic cells. Prokaryotic cells have a variety of shapes. The three most common shapes are spheres or cocci, rods or bacilli, and spirals. Cocci, singular coccus, are spherical prokaryotes. They occur singly, in pairs, diplococci, in chains of many cells, streptococci, and clusters resembling bunches of grapes, staphylococci. Bacilli, singular bacillus, are rod-shaped prokaryotes. They are usually solitary, but in some forms the rods are arranged in chains, streptobacilli. Spiral prokaryotes include spirilla, which range from comma-like shapes to loose coils, and spirochetes, shown in the figure here, which are corkscrew-shaped. Cell surface structures. The cell wall maintains cell shape, protects the cell, and prevents it from bursting in hypotonic environment. Cell walls of eukaryotes are made of cellulose, as in plants and algae, or chitin, as in fungi. Most bacterial cell walls contain peptidoglycan, a network of sugar polymers cross-linked by polypeptides. Archaea contain polysaccharides and proteins but lack peptidoglycan. Therefore, peptidoglycan is found only in bacterial cell wall. Scientists use the Gram stain to classify bacteria by the cell wall composition. Gram-positive bacteria have simpler walls with large amount of peptidoglycan. Gram-negative bacteria have less peptidoglycan and an outer membrane that contains lipopolysaccharides. Look at this figure. You can see here the peptidoglycan layer in the cell wall, thick one, compared to gram-negative bacteria. You can see the thin layer of peptidoglycan, and on top of this peptidoglycan layer, we have outer membrane, and the membrane surface contains lipopolysaccharides. To do gram staining, samples are first stained with crystal violet dye and iodine, then rinsed with alcohol and stained with a counterstain red dye called safranin that enters the cell and binds to its DNA. The structure of bacterium cell wall determines the stain response. In a gram-positive bacteria, those bacteria have a thick wall made of peptidoglycan. The crystal violet enters the cell where it forms a complex with the iodine in the stain. This complex is too large to pass through the thick cell wall, so it is not removed by the alcohol rinse. Therefore, the result, the darker crystal violet dye masks the red safranin dye, so bacteria appears violet blue. Gram-negative bacteria have a thin layer of peptidoglycan, which is located between the plasma membrane and the outer membrane. The crystal violet iodine complex can pass through this thin cell wall and hence is removed by the alcohol rinsed. Result, the safranin dye stains the cell pink or red because crystal violet is easily rinsed. Gram staining is a valuable tool in medicine for quickly determining patients' infection is due to gram-positive or due to gram-negative bacteria. The lipopolysaccharides in the wall of gram-negative bacteria are toxic, causing fever or shock. Furthermore, the outer membrane of gram-negative bacterium helps protect it from body's defense and also tend to be more resistant than gram-positive species to antibiotics because of the outer membrane impedes entry of the drugs or antibiotics. However, certain gram-positive species have virulent strains that are resistant to one or more antibiotics. Example, Staphylococcus aureus or MRSA, which can cause lethal skin infections. Many antibiotics target peptidoglycan and damage bacterial cell wall. Gram-positive bacteria are particularly susceptible to this type of antibiotics. For example, penicillin inhibits peptidoglycan cross-linking. The result cell wall may not be functional, so penicillin destroys many species of pathogenic bacteria without adversely affecting human cells. The cell wall of many prokaryotes is surrounded by a sticky layer of polysaccharides and proteins called capsule. It's present in some of the prokaryotes. The capsule allows adherence to the substrate or other individuals and can shield pathogenic bacteria from the host's immune system. You can see here, this is the cell wall, and above the cell wall we have the capsule, and this bacterium is sticked to tonsil cell in the figure. You can see it here very clearly. Other prokaryotes form metabolically inactive endospores, which can remain viable in harsh conditions for centuries, like Bacillus anthracis, shown below, the bacterium that causes the disease anthrax, الجمر الخبيث اسم المرض, very dangerous. Certain bacteria develop resistant cells called endospores when they lack water or essential nutrients. The original cell produces a copy of its chromosome and surrounds that copy with multilayered structure, forming the endospore. Water is removed from the endospore and its metabolism halts. The original cell then lyses, releasing the endospore. Most endospores are so durable that they can survive in boiling water. Killing them requires heating lab equipment to 121 degrees centigrade under high pressure. Those equipments are called autoclaves. Some prokaryotes have hair-like appendages called fimbriae that allow them to stick to their substrate or other individuals in a colony. For example, the bacterium that causes gonorrhea, مرض السيلان, a sexually transmitted disease, Neisseria gonorrhea, uses fimbriae to fasten itself to a mucous membrane of its host. Pili, or 6 pili, singular 6 pillas, are longer than fimbriae and allow prokaryotes to exchange DNA during the process of conjugation. We're going to talk about conjugation later on. The pillas is a flexible tube of protein subunits. Motility. About half of all prokaryotes exhibit taxes, the ability to move toward or away from a stimulus. For example, chemotaxis is the movement toward or away from a chemical stimulus, such as nutrients. Flagella are the most common structures used by prokaryotes for movement. Flagella may be scattered about the surface or concentrated at one or both ends of the cell. The flagella of prokaryotes and eukaryotes differ in structure, mechanism of propulsion, and molecular composition. They are 1/10 the width and typically are not covered by an extension of plasma membrane. This is the prokaryotic flagella. As you can see, made of motor. This part is embedded in the cell membrane and cell wall, and the other part hook and the filament. Can you tell me whether this bacteria gram positive or gram negative? Think about this. The answer is gram negative. Why? Because as you can see here, you have small, thin peptidoglycan layer. On top of this layer, we have another cell membrane with lipopolysaccharides, as you can see it clearly. Internal organization and DNA. Prokaryotic cells usually lack complex compartmentalization, and we don't have membrane-bound organelles, as you know. Some prokaryotes do have specialized membranes that perform metabolic functions. These are usually enfoldings of the plasma membrane. Look at this aerobic prokaryote. We are going to talk about specialized membrane of prokaryotes, enfolding of the plasma membrane function in serial respiration, similar to the cristae of the mitochondria. Of course, prokaryotes have no membrane-bound organelles, but the equipment needed for serial respiration, like enzymes, like coenzymes, like electron transport chain, are embedded in these enfoldings of plasma membrane. Photosynthetic prokaryotes called cyanobacteria have thylakoid membranes, much like those in chloroplasts. Those are specialized membranes in prokaryotes. Those membranes contain enzymes, coenzymes, photosynthetic pigments, and electron transport chain, and so on. The prokaryotic genome has less DNA than the eukaryotic genome. Most of the prokaryotes genome consists of circular chromosome. The chromosome is not contained in a nucleus. It is located in the nucleoid region with no surrounding membrane. Typical prokaryotes also have smaller rings of independently replicating DNA called plasmids. Most plasmids carry only few genes. See this figure? Notice the single chromosome, and here the plasmid are small rings in this case. There are some differences between prokaryotes and eukaryotes in DNA replication, transcription, and translation. These differences allow people to use certain antibiotics, such as erythromycin and tetracycline, to inhibit bacterial growth without harming themselves. For example, prokaryotic ribosomes are slightly smaller than eukaryotic ribosomes and differ in their protein and RNA content. So these antibiotics block protein synthesis in prokaryotes but not in eukaryotes. This is a summary of the key concepts we talked about. Note here the fimbriae, the cell wall, capsule, here circular chromosome, pilus, flagella, and inside the cell many ribosomes are there, plus the chromosome, which is circular and are contained within nucleoid region, and we can see a small circular DNA called plasmids. Reproduction. Prokaryotes reproduce quickly by binary fission and can divide every 1 to 3 hours under optimal conditions. Key features of prokaryote biology include: first, they are small. They reproduce by binary fission, making colonies. They have short generation times. They produce a large number of offspring cells. Now, we're going to talk about the second concept that deals with diversity in prokaryotes. This concept is entitled rapid reproduction, mutation, and genetic recombination promote genetic diversity in prokaryotes. Prokaryotes have considerable genetic variation. Three factors contribute to this diversity: rapid reproduction, mutation, and genetic recombination. Genetic recombination involves three mechanisms: transformation, transduction, and conjugation. Rapid reproduction and mutation. Prokaryotes reproduce asexually. Offspring cells are generally identical. For example, Escherichia coli cells reproduce using binary fission. Mutation rates are low, 1 in 10 million per cell division. But mutations accumulate rapidly because generation times are short and populations are large. The key point is that new mutations, though are rare on per gene basis, can increase genetic diversity quickly in species with short generation time and large populations. This diversity, in turn, can lead to rapid evolution. So, prokaryotes are highly evolved organisms. Now, we're going to talk about genetic recombinations, the sources of genetic variation, since prokaryotes have no sexual reproduction, and sexual reproduction in eukaryotes involves meiosis and so on. Therefore, we'll see the sources of genetic diversity in prokaryotes. Prokaryotic DNA from different individuals can be brought together by different mechanisms: transformation, transduction, and conjugation. Movement of genes among individuals from different species is called horizontal gene transfer. Now, we're going to talk about transformation. A prokaryotic cell can take up and incorporate foreign DNA from the surrounding environment in a process called transformation. The genotype and possibly the phenotype of prokaryotic cell are altered. For example, harmless strain of Streptococcus pneumoniae can be transformed into pathogenic strain by uptake of DNA from heat-killed pathogenic strains. Remember this in chapter 16. This is the experiment of Griffiths. The pathogenic cell is now recombinant cell. Its chromosome contains DNA derived from two different cells. The other source of genetic variation in bacteria, transduction. Transduction is the movement of genes between bacteria by phages or bacteriophages, viruses that infect bacteria. Phages may carry pieces of bacterial chromosome from one cell, the donor cell, to another, the recipient cell, if crossing over occurs after the transfer of genes from the donor may be incorporated into the recipient's genome. See figure in the next slide showing the replicative cycle, the lytic cycle of the virulent phage. This slide shows the lytic cycle of phage T4, a virulent phage. This phage, its genome is DNA, as you know. First step, when the phage attack its host, bacterial host, like E. coli in this case, the first step is attachment. After attachment, entry of the phage DNA and degradation of the host DNA. Then, the start of synthesis of viral genome and proteins, because the phage DNA directs production of its own proteins and copies of its genome by the host and viral enzymes using components within the host cell or the infected cell. After that, we reach the stage of self-assembly. These parts of the viruses, composed of sets of proteins, will self-assemble to form phage heads, tails, and tail fibers. Those are details not important for you now. The phage genome is packaged inside the capsid as the head forms. Then, before the release, the final step, the phage directs the production of an enzyme that damages the bacterial cell wall, allowing fluid to enter into the cell, no protection. The cell swells and finally bursts, releasing 100 to 200 phage particles. So now we're going to explain transduction based on the life cycle of this phage. Then, in this figure, showing transduction process, phage infects bacterial cell, the donor cell, that carries the A plus and A and B plus genes or alleles. Phage DNA replicated and phage proteins are synthesized. Then, as new phage particles assemble, a fragment of bacterial DNA carrying, say, the A plus allele happens to be packaged in the phage capsid or a B plus allele. The phage carrying the A plus allele from the donor cell infects a recipient cell with allele A minus and B minus. Crossing over at two sites allow donor DNA to be incorporated into recipient DNA. Finally, incorporation of phage DNA creates recombinant cell with genotype A plus B minus. Now, we're going to talk about conjugation and plasmids. Another source of genetic variation in bacteria is conjugation. Conjugation is the process where genetic material is transferred between prokaryotic cells. In bacteria, the DNA transfer is always one way, from one cell, donor cell, to another cell, recipient cell. A donor cell attaches to a recipient cell by a pilus, pulls it closer, and transfers DNA through a structure called the mating bridge. A piece of DNA called the F factor is required for the production of pili. The F factor as a plasmid. Cells containing the F plasmid function as DNA donors during conjugation. Cells without the F factor function as DNA recipients during conjugation. The F factor is transferable during conjugation. Provided some of the F plasmid's DNA is transferred, the recipient cell becomes a recombinant cell. Now we'll see this figure talking about conjugation and recombination in Escherichia coli. A cell carrying an F plasmid, an F plus cell, forms a mating bridge with an F minus cell. One strand of the plasmid's DNA breaks at the point marked by the arrowhead, as you see here. Using the unbroken strand as a template, the cell synthesizes a new strand, light blue color. Meanwhile, the broken strand peels off, red arrow, see here, and one end enters the F minus cell. Their synthesis of its complementary strand begins. DNA replication continues in both the donor and recipient cells as the transferred plasmid strand moves further into the recipient cell. Once DNA transfer and synthesis are completed, the plasmid in the recipient cell circularizes. The recipient cell is now a recombinant F plus cell. Are plasmids and antibiotic resistance? Are plasmids carry genes for antibiotic resistance? Antibiotics kill sensitive bacteria, but not bacteria with specific R plasmids. Through natural selection, the fraction of bacteria with genes for resistance increases in population exposed to antibiotics. Many R plasmids have genes that encode pili, making it possible for resistance genes to be transferred between bacterial cells during conjugation. Some R plasmids carry genes for resistance to multiple antibiotics. Antibiotic-resistant strains of bacteria are becoming more common, and the infections they cause are now harder to treat. To know more about antibiotic resistance and what causes antibiotic resistance, watch these two simple explanatory videos. Both links are available in this video description below.